I'm very sorry to not be there in person. I really missed this meeting and was hoping I could. So let me see if I can share my screen. There we go. So I am going to be talking about what exactly we mean by phage sensitivity and how we should detect it in personalized phage therapy, specifically in routine practice. So this is a slide that I think this audience doesn't really need. So I'm just going to breeze right on past it. I hope I'll be able to ask for some ability to introduce what we're doing, because it's somewhat different from what many others are doing. So just to briefly describe what we're not doing, the conventional medicinal product development pathway. Now, in this pathway, at least as it would be adapted to phage therapy, a phage cocktail would be designed with enough phage to be able to plausibly have at least one phage that would infect any given clinical isolate of a specific species, or at least a broad host range of potential clinical isolates that a patient might be suffering from. This cocktail would be, each member of this cocktail would be manufactured as an active pharmaceutical ingredient according to GMP, formulated into a cocktail according to GMP, and then tested in preclinical testing, and then tested for safety, and then tested for efficacy in humans. Assuming there are positive results, the platform would then be reviewed and hopefully approved by a competent authority to receive marketing authorization, and then move on to phase 4 pharmacovigilance safety testing during routine use. However, we see this as not being a pathway that's at least immediately available for phage therapy, particularly given the challenges of the antimicrobial development space, where this is essentially the same pathway that's been used for quite a few failed antibiotic designs, and we see that phage is not really adding anything new so long as the framework stays the same. Where with phage, they induce resistance to phage resistance far more quickly than antibiotics induce antibiotic resistance. And so by the time a phage cocktail would receive marketing authorization, that'd be a 10-year and billion-dollar process. But potentially the phage might not be relevant at the end of those 10 years. But even then, the more the phage is used, the more it selects for the resistance to the phage in the cocktail, the less the phage are useful, creating the same dynamic that has prevented the development of new antibiotics, as physicians would be obliged to exercise phage stewardship in exactly the same ways that they've been obliged to exercise antibiotic stewardship. As physicians would use the phage less and less in order to conserve the activity, fewer and fewer patients would be paying for that phage, leaving the cost of development to be laid onto fewer and fewer shoulders, to the point where eventually it would simply not be accessible anymore. We see a fundamentally different framework as being appropriate for phage therapy, similar to the dream that Ben Temperton discussed in his excellent talk about the Citizen Phage Library. This would be the traditional approach to developing pharmaceuticals, where phage would be produced in a tailor-made, personalized fashion. So in this model, we pre-manufacture a regularly updated bank of quality-controlled, small-scale produced phage stocks. These are then selected from using a laboratory diagnostic to identify which phage is appropriate for a specific patient. And then only the phages that have been identified as being appropriate for a specific patient, the phages that propagate at the expense of the patient's specific clinical isolate, are then formulated into whichever form the patient happens to need, whether that's a bladder wash or a cream or a preloaded syringe, and then it's applied on the basis of a prescription to a patient. such that then when phage resistance ensues, the clinical isolates that are resistant to all of our phage can then be brought back to our laboratory to be used as tools to isolate or train new phage that can then go back into the library, creating a system that is sustainable. Because the thing that we're investing in isn't the medicinal product that we're developing, not exactly. As that medicinal product loses relevance, that's fine for the model. The thing that we're really investing in is the laboratory diagnostic, which then has the ability to recommend phage that are safe and efficacious for a specific patient. So, really, we see this one-size-fits-all approach of fixed phage cocktails that don't evolve as being at least not an initial step, a first step for the phage therapy field, particularly because of the high failure rate in clinical trials. There have now been 4 clinical trials, all of which have failed to cleanly demonstrate efficacy, all of which used functionally fixed models. But there are naturally advantages. There'd be the possibility to protect a specific cocktail. And it would be substantially more accessible. There'd be no need for that laboratory diagnostic between the patient and their treatment. But we see the personalized medicine model as being the appropriate first step for phage therapy, specifically since the personalized approach includes that diagnostic step that determines the optimal phage selection, where this approach can sustainably adapt to changing epidemiological circumstances. It has substantially reduced formulation challenges and allows for the substantially expanded pharmacovigilance that's really necessary for a field with our challenges, where we need a much more flexible approach to assessing efficacy. But the biggest reason why we see this as being necessary is that almost all of the compelling evidence for phage therapy efficacy has been generated through at least functionally personalized models. So what this requires, though, is a phagogram, a rapid phagogram, because historical methods for determining phage susceptibility are just fundamentally inadequate to the task at hand. Where it might be adaptable to the patients who are coming to us, but for every patient who's able to find phage therapy and access phage therapy, there are thousands more who really need us to come to them. And neither plaque assays nor spot tests nor liquid assays are able to really do that. They require far too much labor. The work hours spent per test are just fundamentally unacceptable for routine implementation. And they require substantially trained personnel that are just not available in local clinical microbiology labs. They require far too much time between the collection of the patient sample and the provision of the diagnostic result. Realistically, a 72-hour delay is already too much, and that's what's needed at the moment for an antibiogram to be performed to direct antibiotic therapy. At the moment, more than 90% of antibiotics are prescribed empirically. And a 72-hour delay is quite a challenge to meet already. So for phage therapy to be really feasible in routine, a physician really has to have an actionable answer in no more time than it takes for an antibiogram. But perhaps the biggest reason why they can't really be made to work is they're difficult to consistently interpret. Does a turbid plaque or a turbid spot indicate sensitivity or resistance? How small can a plaque really be and still be relevant? If you show one plate to 5 phage biologists, you're liable to get 8 different answers as to what that plate means. And that doesn't work in a professional environment. So for a phagogram to truly be implementable in routine therapy, it needs to be feasible, including in every small laboratory hospital where patients currently access an antibiogram to direct antibiotic therapy. And it can't require expensive disposables or equipment. It can't require specialized academic labor. It can't be bulky. The thing needs to be user-friendly enough to train someone on within a few hours. it needs to be fast, with a turnaround time that's no longer than for antimicrobial susceptibility testing. It obviously needs to be highly sensitive and highly specific to be a useful diagnostic. And the standardized results need to be clear and easily understood by clinicians, as is currently mostly the case for antibiotics. And this is really what I want to focus on today. Our vision for what this really means. So what does susceptibility mean? No, what does it mean for a bacteria to be susceptible to a clinical isolate? Well, there's a functionally infinite number of possible relationships that a clinical isolate can have to any given phage. No, but we see a need to summarize these relationships into clear categories. And we don't think that susceptible and resistant are enough. No, where there's also particularly abortive infection, where in the process of phage infection, after attachment, for the most part, in almost every case, that bacteria is dead. The moment it has a phage successfully attached to it, that bacteria is not going to survive as a colony-forming unit. But that doesn't necessarily mean that that phage-infected cell is going to produce phage progeny. And a substantial fraction of the possible relationships between a given clinical isolate and a therapeutic phage, that interaction will result in what's called abortive infection, where some downstream aspect of the infection is disrupted through some means, either through a phage defense mechanism like Franklin Nobrega talked about in the talk just before mine, or because the host just happens to be missing something that the phage happens to need. No, the cell dies, but doesn't produce phage. So in that context, what we'd have is a PK/PD relationship that'd be a lot more recognizable to most physicians, where just like for most other antimicrobial therapies or therapies in general, the physician would be able to apply the phage to the patient. It would then reach the minimum effective concentration, at which point it would begin to act. It'll reach a peak density, hopefully below the minimum toxic concentration, and then begin to be excreted through either metabolism or dilution before it reaches the minimum effective concentration again, at which point the effective therapy ends. So a phage that produces abortive infection on its target will be somewhat similar to this. It's just a therapy that would be that would kill itself in its activity, which is also relatively normal. But this isn't really what physicians are expecting from phage. What they're expecting is the story that we as a community have been telling for the last 20 years, which is that phage will do something more like this, where the phage are applied, they get diluted and inactivated just through diffusion into the body. But at a certain point, the phage will find its host and begin to replicate in situ, at which point it will begin to, the concentration will begin to go back up, up to the minimum effective phage density, allowing the infection to clear, regardless of how many phage actually reach the site of infection. But this requires the phage to be able to actually productively infect the bacteria, which is something that in personalized therapy, we think is going to be really necessary, such that we can be clear to physicians about what exactly they can expect from the phage that our diagnostic will be recommending. And so just understanding this through the reference methods, the Gratia method and the spot test, we see a propagative phage as being able to form clear, large enough plaques with a high enough EOP on the clinical isolate. Whereas we see a room for another diagnostic category of active, where that would reflect some form of abortive infection, where in the reference methods, this would be a clear spot without plaques. This would be very small plaques where the replication potentially is there, but might not be clinically relevant, or with a very low EOP that might reflect some replication, but not any that could be plausibly clinically relevant. and then a variety of possible biological relationships that would then be insufficiently sensitive for us to recommend this phage for therapy. So how did we develop this phagogram? Oh, well, the phagogram that we've designed. So we started from first principles. Oh, in each of these 5 steps of phage infection, there's some part of it that would be vulnerable to detection, where what we want to do is detect the phage infecting or fail to detect the phage infecting to be able to then determine whether our phage infects a specific clinical isolate for a patient. And our first thought was, oh, let's just detect the arrestive cell growth. That should be relatively easy. Where the core challenge of developing a phagogram is just the very limited number of cells that are available to detect. So what if instead of detecting phage infection, we instead detected the growth of uninfected cells? So instead of detecting susceptibility, we detected resistance and inferred susceptibility from the absence of resistance. So we have an omnilog, and we could have developed this phagogram, but we decided not to. Where we figured that this would be a trap, where it could only be performed on pure culture clinical isolates, because any contaminating bacteria in there would then be a resistant bacteria that we'd infer the absence of susceptibility to. It requires an additional 18 hours, and every hour really counts for many patients. It'd require a bulky and expensive robot, which would functionally necessitate a small number of core facilities, which would mean that when a patient sample is taken, it'd need to be turned into a pure culture clinical isolate in their local lab. That isolate would then need to be shipped to us. We would then need to turn it into a new pure culture clinical isolate. We'd then need to put it into our robot. And each of these steps takes another 24 hours. And particularly with shipping, what we'd really be talking about would be a minimum lead time of 2-3 weeks at best. Functionally a minimum of a week, but this is a professional environment. People go home at night. They have weekends. This isn't an academic environment anymore. And so what we decided to do instead is focus on step 5, the lysis and release of cell contents. So just starting from first principles, what if we were to detect the release of cell contents rather than the cells themselves? Inside every living bacterial cell is ATP that would be available to power a luciferase enzyme. So we put into the system luciferase and luciferin, which with oxygen turns into light. And quite a bit of light, which can be quite sensitively detected by a camera, quite automatically. And these assays can be extraordinarily sensitive. So most healthy bacterial cells contain somewhere around 2 × 10⁸ moles of ATP per cell. And with our limit of quantification, we can detect between 5 and 200,000 healthy bacterial cells. And that's more than low enough to be able to divide a pure culture clinical isolate however many times we need to, in order to make however many tests we need to, in order to make an effective test against hundreds or even thousands of phage. So let me show you some of our results. So here on the left is a plate made using the reference method. This is just a spot test. And it's a plate that if you were to show it to 5 phage biologists, I think you'd really get one answer as to what it means. Phage red and phage blue are each producing nice, beautiful plaques, clear, beautiful lytic spots, indicating susceptibility, true susceptibility, that they propagate at the expense of this clinical isolate, and also that they kill this clinical isolate quite effectively, whereas phage green isn't doing anything. And so just feeding these phage into our robot, we can see that phage red and phage blue each release ATP after about an hour, hour and a half, and phage green does not. But let me show you a plate that if you were to show it to 5 phage biologists, you might get 10 answers as to what it really means. So here, phage red is producing big, beautiful plaques, and very clear, beautiful lytic spots. But phage green is producing maybe a turbid spot. Those might be plaques in there. It's not really clear. And phage blue is producing sort of a small, sickly, but clear lytic spot. What does that really mean in terms of the relationship between these phage and this patient's clinical isolate? Well, our phogogram gives, I think, a very clear answer. Where phage red, you can see essentially a two-step growth curve in the release of ATP from the clinical isolate, cells of the clinical isolate. Whereas phage green and phage blue, what you're seeing really is kind of clearly just toxicity. And not very much toxicity.
Sorry, Bob, this is the chair here. I wonder, look, we're just pressed for time, and I wonder maybe could you just go to your last maybe concluding slide? Apologies for this, but we're just up against the wall for time.
This more or less is the concluding slide, so you're all right.
Thank you.
So, by showing in our assay, by showing what percentage of the culture is lysed as a result of phage-mediated activity, we can divide different clinical isolates into 3 different categories, reflecting true phage propagation, phage toxicity, and just an insensitivity to phage, which consistently agrees with the reference methods. And so, yeah, this is what the robot looks like. It's about the size of 2 toasters, and works quickly and can be implemented anywhere an antibiogram is implemented, allowing it to be accessible really to patients at scale. And so with that, I'd like to just quickly thank our partners at the Laboratory of Brussels University Hospitals, who have been doing all of the real hard work of developing this method, and take questions if there's time for them.
Lovely. Thank you, Bob. Thank you for that presentation. So slightly potentially disruptive paradigm for personalized medicine in the age of phage therapy. Have we any questions for Bob? Just one thing, Bob, on the personalized use of phage and the delivery to the particular, we'll say, target organ or target area, and the amount of phage that's needed. How are you going to kind of, we'll say, make sure you have sufficient phage to address the, we'll say, bacterial load or bacterial burden on the particular organ or tissue?
So this is a decision that I think that I'm not a physician, and I think this is a decision that will need to be made by physicians. And so we plan to make 10 to the 10th plaque forming units available in a vial to hospital pharmacies to then be formulated in whichever form and whichever concentration a physician decides. So we're developing a committee of physicians who will be able to advise new physicians as to what would then be the most appropriate formulations for a given patient, given their specific clinical context.
So we'll have a new branch of medics coming out, those that are familiar with all this language that we're talking about today. Okay, well, if there are no more questions, thank you very much, Bob, for your very interesting presentation.
Thank you.
Thank you.